Welcome to the first module of Biosafety training at UW-Milwaukee. Module 1, an introduction to biosafety, will introduce you to core concepts of biosafety at UW-Milwaukee, and an overview of the regulations and guidelines you are expected to follow when conducting research or teaching experiments involving biohazardous materials at UWM. What is biosafety? Biosafety combines knowledge, techniques, and equipment to prevent personnel, public, and environmental exposure to potentially infectious biohazardous materials. The University of Wisconsin-Milwaukee Department of University Safety and Assurance's Biosafety Program oversees the responsible use of biological hazards in microbiology, tissue culture, recombinant DNA, molecular biology, synthetic biology, and biotechnology at all the UWM facilities. The Biosafety Officer, or BSO, and the Institutional Biosafety Committee, or IBC, evaluate and approve protocols for research experiments and teaching experiments that work with biological hazards. The UWM Biosafety Program supports research and teaching at our institution through the following means. We promote safe science when handling biohazardous materials, including the practices, procedures, and containment equipment used in your laboratories. Our program provides the biosafety training for personnel at UWM regarding the safe and ethical use of biological materials in research and teaching. We consult with facilities, architects, building engineers, and faculty on lab design and biocontainment equipment. The BSO acts as the administrator for the Institutional Biosafety Committee and coordinates meetings, protocol review, and acts as a liaison between faculty and the IBC. We support teaching and research efforts at UWM through excellent customer service and guidance regarding biosafety and University Safety and Assurance's program offerings. The following are the roles and responsibilities of principal investigators, laboratory managers, and supervisors in laboratories handling biohazardous materials. It is required that you possess the appropriate training and education to handle the biological materials being handled in your lab. It is your responsibility to ensure the laboratory is safe, organized, and personnel are appropriately trained and educated in the work they are performing. It is your responsibility to manage biosafety protocols and submit them as necessary for biosafety and IBC review. It is the responsibility of the leaders of the lab to maintain standard operating procedures, biosafety manuals, and any other important information necessary to provide a safe working environment. It is the responsibility of the leaders to schedule biosafety lab visits and remediate issues that may arise from those visits. It is also your responsibility to maintain a biohazard spill kit in your laboratory, as well as a clear biohazardous spill standard operating procedure. Now let's turn to the roles and responsibilities of research and teaching personnel. It is your responsibility to possess the appropriate training and education to handle the biohazardous materials being utilized in your laboratory. It is your responsibility to be responsible for your work and maintain a safe and organized workspace. You must submit biosafety protocols on behalf of your principal investigator and obtain approvals prior to commencing experiments. You need to make sure that you are regularly reviewing, understanding, and following all laboratory standard operating procedures and biosafety manuals. It is your responsibility to adhere with local, state, and federal guidelines and regulations regarding biohazardous materials. Report any unsafe practices or near misses, as well as any accidents and exposure events that involve biohazardous materials. You should know the location of your biohazardous spill kit and the standard operating procedure to follow in the event of a biohazardous spill. It is also your responsibility to conduct daily risk assessments prior to conducting experiments to reduce risk in the laboratory. Teaching personnel have unique roles and responsibilities when working with biohazardous materials with their students. They need to be responsible for reviewing all teaching activities and identifying the risks associated with the teaching activities beforehand so they can use this to effectively communicate those risks and find ways to mitigate risk with their students prior to commencing any activities. They need to ensure that the laboratory is safe and organized. If you're teaching in a teaching laboratory that involves the work with biohazardous materials, it is your responsibility to convey the appropriate biosafety information to students prior to commencing the teaching lab activity. Enforce a dress code, a cell phone use policy, PPE, and safe lab practices guidelines in your classroom. In addition to biosafety training, you may be required to complete additional training specific to your research area. If you are looking for guidelines for safe practices in biosafety at the University of Wisconsin-Milwaukee, you may visit our UWM Biological Safety Manual website to download the UWM Biosafety Manual, the Biosafety Guidance page, which will provide you with resources for work with biohazardous materials, or the IBC webpage, which will lead you to submission of biosafety protocols as well as any other important information you need for research compliance in biosafety. In addition to biosafety, you can find out more information regarding lab safety, radiation safety, animal care, human subjects research, and more by visiting our website at www.uwm.edu/USA. What are biohazardous materials? Biohazardous materials are biological materials that pose a potential risk to humans, animals, and/or the environment. The containers and equipment used to manipulate or store these materials must possess a biohazard symbol, as seen here in the photo. Let's take a look at biohazardous materials. They may include bacteria, such as Staphylococcus aureus, as seen in this photo, as well as the toxins bacteria may produce, viruses, such as the Ebola virus, as seen in this photo, or other viruses that are commonly used in research, such as viral vectors, algae, such as the blue-green algae responsible for algal blooms in lakes around Wisconsin and beyond, and their toxins that they produce, prions, which lead to spongiform encephalopathies, such as bovine spongiform encephalopathy or mad cow disease, parasites, such as the tapeworm in this photo, and fungi, such as the candida or yeast shown in this picture. Besides microbiological agents, human and non-human primate cells, fluids, and tissues are considered biohazards, including cell lines and human blood samples. Animals that are used in biological research, such as rodents for disease models or use of transgenic organisms, are also handled as biohazards. This also includes all arthropods. If it belongs to the kingdom Animalia and you plan on manipulating it or infecting it, it becomes a biological hazard. Shown here is a lab mouse and zebrafish, two commonly used animal models in research. Biological toxins are produced by animals and bacteria they are both a chemical and biological hazard. Two examples shown here are those that are produced by the black widow spider, which produces the neurotoxin alpha-latrotoxin, and cone snails, which produce conotoxins. We also handle exotic or invasive plants as biohazards, as we seek to ensure that they are completely destroyed prior to disposal and that we contain their pollen and seed, as they are risks to the environment. Shown in the photos are purple loosestrife and garlic mustard. All modified organisms are handled as biohazards. Shown here are zebrafish that express the green fluorescent protein, making them bioluminescent. Nanomaterials used in biological systems or agents are both a chemical and biological hazard. In addition to animal biological toxins and other biological toxins produced by bacteria, plant toxins are also of concern. Shown here is a castor plant in which the seeds possess ricin, a potent toxin. The NIH and WHO recommend 4 risk groups based on the following hazardous characteristics of an agent: its ability to infect and cause disease in a susceptible human or animal host, its virulence as measured by the severity of the disease, and the availability of preventative measures available and effective treatments available for the disease. The risk group listing from the NIH guidelines or the Biosafety and Microbiological and Biomedical Laboratories are the current standard used for risk group classification. Risk group 1 agents typically are not associated with disease in humans. Some examples may include Staphylococcus epidermidis and K12 strains of Escherichia coli. They have a low individual risk and a low risk to the community. Risk group 2 agents are associated with human diseases that typically have preventative or therapeutic interventions available, such as antibiotics or vaccines. Examples include Staphylococcus aureus, Salmonella species, Enterotoxigenic E. coli, and many others. Many agents handled here at UWM associated with human disease fall into a risk group 2 classification. There is a moderate risk to individuals coming in contact with these pathogens, but a low risk of it creating a community issue, so the likelihood of spread from person to person is fairly low. Risk group 3 agents are agents associated with serious or lethal human diseases that may or may not have preventative or therapeutic interventions available. We do not work with risk group 4 agents at all at this university, but those are agents that would be very likely to cause serious or lethal human disease for which there are not really any preventative or therapeutic interventions available, and have a high risk to both individuals and the community. These would be things like the Ebola virus that require very high containment. Biosafety levels and risk groups do not go hand in hand. Contrary to that popular belief, you may have an agent that is very low risk to humans or animals, but it could be of serious consequence to the environment. So we have to take into consideration the biosafety level in which you work in to handle the agent that you plan on studying. The biosafety level defines the type of containment, the equipment, the personal protective equipment, and the laboratory practices implemented to actually work with a biological agent. BSL-1 laboratories would be the lowest risk, or the lowest level of containment, and BSL-4 would be the highest level of containment. This concludes Module 1. To continue the biosafety training, complete the short follow-up quiz. You must score 80% or higher to move on, but you have unlimited attempts to complete the quiz. Module 2 will focus on risk assessment.